let's talk about proteins. Now, most of the time when you think about proteins, you think of people with really big muscles, like this guy down here. But that's not always the case. Yes, muscles are made out of protein, but there's a lot of other things that we need to think about as well. So, yes, a lot of meats, a lot of fish, a lot of beans, eggs, things like that, they do have protein. But things that you may not think about, spider webs. Spider web is actually pure protein. Feathers, hooves on animals, the antlers or the horns on certain animals, hair, skin, nails, those are all proteins. Actually, proteins make up pretty much over 50% of what's ever in your body. Everything is protein based. And so let's look at the characteristics of a protein. Of the schnapps, proteins are comprised of everything except for phosphorus. So they have carbon, hydrogen, oxygen, nitrogen, and sulfur that make them up. They are the most abundant macromolecule that there is, making up over 50% of your total mass. And so they're super important. So let's look at their structure. Their structure is actually very unique. Every single protein has a general structure, but the protein that's actually formed is a little bit different from every other protein that's inside of your body. So each protein has a unique structure, giving it a very unique function. Everyone does a little bit different, has a little bit different of a job. These are different examples of places that you may think of proteins that I just talked about. Anything from enzymes that speed up chemical reaction to antibodies that fight off disease, your skin, hair, nails, muscles, ligaments, hormones, the spider webs, the feathers, blood clots, everything inside your body is actually protein based. So let's look at the structure of amino acids, which is the basis of all proteins. So the monomer of a protein, the monomer, the smallest unit, is called an amino acid. There are actually 20 different types of them. And I'm going to show you the chart eventually sometime in the year, but you don't need to know it right now. But glycine, isoleucine, valine, tryptophan, aspartic acid, leucine, there's 20 different types. But each one of them is considered a monomer, a single unit of a protein. So you have a picture that looks something like this, not quite the same, but each one of these circles is one amino acid. When we link a bunch of them together, we get something called a polypeptide. So the polypeptide is the actual polymer of a protein. It's when the many monomers are joined together. And the reason it's called a polypeptide is because between each one of these amino acids, there's a special type of covalent bond. And that covalent bond is called a peptide bond. So if you have a lot of peptide bonds, that's where it gets its name, polypeptide. We're not done there yet. When you have a bunch of polypeptides, you actually don't have a protein quite yet. You actually have to get lots of polypeptides together. So this red polypeptide, let's pretend it's this red one down here. Okay. Notice that there's a green one, a blue one, a yellow one, a purple one, a darker blue colored one. These are many different polypeptides joining the red one. So a protein is when many of these polypeptides or these chains actually get together. They bond, they fold, they twist, they look like this ribbony confetti sort of stuff. They will have a specific structure that gives them a specific function. Let's look at the amino acid structure. Amino acids have a general structure, but there's 20 actually different versions of them. But all of them look like this. They have a central carbon, and off of carbons make 4 bonds. So off of the one side is going to be hydrogen, and then off of one side you're going to have something called a carboxyl group. So this blue box right here is called a carboxyl group. Should sound familiar. They're in fatty acids. They're the carboxyl head of a fatty acid. COOH. COOH. Over here, on the other side, in this green box, we have something called an amino group. An amino group is pretty much a nitrogen and 2 hydrogens. So NH2 is an amino group, carboxyl group is COOH. And then the last part of the amino acid is right down here. This is called the side group, also known as the variable group, because it varies, because it could have 20 different versions. There's 20 different amino acids. So if I look at this picture, there's my amino group, it doesn't change. Here's my carboxyl group, it doesn't change. I have my central carbon and my hydrogen, just like I do up there. The only thing that changes or varies is this side group, or the R group. So the R group in alanine is a specific amino acid. This is what its structure would be. But if I change my amino acid to serine, then its R group would change potentially great or slightly. This one, it's only different by an oxygen. And so Each one has a slightly different R group, which makes each amino acid slightly unique. When we take two amino acids and we put together, no matter what two of the 20 we put together, two of the same or two different, here's one amino acid and here's the other, you could see here's my amino group, here's my carboxyl group, and my side group. That's what they all have. But to put them together, you have to do dehydration synthesis. So off the carboxyl group, you have to take an OH, and off the amino group, you have to take a H, and there goes my H2O. It's actually shown right there going out. And that will leave the carbon and the nitrogen to form a bond between each other. And that type of bond is a covalent bond called a peptide bond. And when you get many of them together, it's a polypeptide. If you want to break down the polypeptides, or in this case there's only two, so it's a dipeptide, you put the water back in and it will break them apart by hydrolysis. So they are all put together by dehydration and they're all taken apart by hydrolysis, just like every other macromolecule that there is. Here's actually the 20 different amino acids. And in college, I had to actually memorize all these and all their structures and all their names and all their abbreviations. And that was not a very pleasant time for me in college, let me tell you. I still have all the note cards that I drew out relentlessly for a long time studying. But they're categorized based on their properties. Some are polar, some are nonpolar, some form acids and some form bases. Don't care that you ever know that. But I want you to notice one thing. The part that's in a darker color, these are all the side groups. These are the only things that are changing in all the amino acids. This part is the amino and the carboxyl groups that are always staying the same, that never changes. Some of the amino acids of the 20, we actually have to have all 20 in our body, but we don't produce all 20 of them. We actually only produce, I believe, 11 of them, which means that we have to consume the other 9 from what we eat. And we need the ones that we need to eat are essential that we eat them, so we call them essential amino acids. But the ones that our bodies produce, it's not vital that we actually eat them because our bodies are already producing, so who cares if we never eat them? They're always there because it's not vital or not essential to eating them because our bodies make them. We call them non-essential amino acids. So essential amino acids and non-essential amino acids. You need all 20 of them, but you don't make all of them. Let's look at the protein functions. The first function of proteins is that they're good at building things. They're good at structures. So your hair, your skin, your nails, they're made out of actually these proteins called collagen, elastin, and keratin. Your hair is mainly keratin. I spelled keratin. It should have a T in there. But elastin makes your skin elasticy, and collagen makes it kind of tough and waterproof. The second type of function is transport, and so you actually have proteins in your blood. One of them is called hemoglobin. It transports oxygen to wherever it needs to go, which is all your cells. Another function is motion. It helps your muscles move, for example. Inside your muscles you have two proteins called actin and myosin. They will actually cause contractions in your muscles that allow for movement or motion. The next one is defense. Your body has to protect itself against viruses and bacteria that can make you ill. So you have these proteins called antibodies that will fight them off. Your proteins store stuff. They could actually store whatever they need to store. In this case, in milk, there's a protein called casein, and that actually stores some of the energy needed for protein development in young mammals and from their mother's milk. There's also one in eggs. So when you look at the egg white of an egg, and everybody knows hopefully that eggs are a good source of protein, and the reason because of that is because of the white of the egg, and it's called ovalbumin, and that's the stuff that you actually stores the protein for the developing embryo. Hormones, such as insulin and glucagon. Insulin and glucagon are two hormones that control blood inside of your sugar. They regulate how much sugar is inside of your blood. You can't have too much, but you can't have too little, because that could cause problems. You also have other hormones, like insulin, not insulin, but testosterone and estrogen, and you have lots of different cholesterol even though we talked about cholesterol being a lipid, it's actually part of its protein as well. Enzymes. We're going to have a whole unit on enzymes. They speed up chemical reactions. So if it ends in ase, it is an enzyme. Lactase breaks down lactose, which is a sugar, the disaccharide. We have protease, which breaks down proteins, and we have lipases, which breaks down lipids. And there's one for everything inside of your body. Buffers, we're going to talk about in class. I'm going to save it for in class. Buffers, as we talked about with acids and bases, they help maintain pH, but we're going to learn how that could actually happen in class. The last thing I want to talk about is protein folding and protein structures. So protein, when you have a bunch of amino acids together, here's a bunch of amino acids making a polypeptide, it needs to actually fold together to make a functional protein. And so it has to go through these different protein folding steps that we call structures. The first one is called primary because it's first, and it's just a straight chain of amino acids put together into a polypeptide. And then we make a secondary structure down here where this straight chain will actually start to fold or bend or twist. And then what will happen, it will start to actually fold more on top of itself. And then after that, it will get together with another polypeptide and make a true protein. So let's look at this a little bit more in detail right here. Here's our primary structure. It's a straight chain of amino acids. It's a polypeptide. That needs to fold. It folds by folding back and forth like a beta, this is called a beta pleated sheet, or it will actually make the spiral sort of thing called an alpha helix. So it will twist or it will fold. And then what happens, the polypeptide will actually fold more on top of each other. You can see that here's the twisting that's still there, but the whole thing is actually folding on top of itself. So it's making this complex 3D structure. That complex 3D structure, or polypeptide, will get together with more complex 3D tertiary structure polypeptides. And so right here, you can see that there's 1, 2, 3, 4 polypeptides that are all folded together in this structure. And so this is where we actually get the functional protein. So it takes a lot for them to fold together to actually become useful and functional. So a protein has to be 2 or more polypeptides folded or coiled together into a specific shape. And then that shape determines the job of what it does. You have a protein for everything different inside your body, and its structure determines what it does. And if it's not shaped that way, it will not work. If it loses its shape at any time, it's called denaturation, or it becomes denatured. That means that it unravels and no longer functions. So hemoglobin, here's the general picture of what kind of hemoglobin would look like as a molecule. We use ribbons to represent how it folds. And it's made out of 4 polypeptides, 2 blue and 2 red in this picture. If it's not shaped exactly just like that, it will not carry blood to all your cells, and that's a problem because you're dead. Being dead is definitely a problem. This one, insulin. If insulin is not shaped that exact same way, 3 polypeptides put together, actually 2 polypeptides, but they're shaped that way, you don't control the blood in your sugar. That's definitely a problem. You're diabetic. If you get bit by a snake and it has a neurotoxin, that's a type of venom that goes inside your body. It's a protein that's being injected inside of your body, and that's a problem. This protein causes your neurons and your muscles not to work. And so you don't want it to be this shape. And so that you actually have to take an antivenom that actually destroys the shape of it so it no longer messes with your muscles and nerves and doesn't kill you. And so they could be kind of both sides there, good or bad. So denaturation is when you lose that shape and therefore it no longer functions. What can do this? Well, if you change the temperature of something, it can denature a protein. If you change a pH, or if you expose it to certain heavy metals, then it could actually disrupt the nature of how it's held together and unravel. And you can see that this one is becoming unraveled or denatured by one of those 3 things. An example of temperature would be cooking an egg. You could actually see the clear mucousy egg white turning to egg white. It's denaturing in front of your eyes. It no longer has its storage function anymore. Ladies, if you straighten your hair with a straightening iron, or gentlemen, okay, now I'm not judging here, but if you have a straightening iron or curling iron, your hair is pretty much pure protein, and you actually temporarily denature it by making it straight. Changing pH, we could talk about that in class as well. So those are some different examples of how you could unravel a protein so it no longer works. I hope this was helpful. There's a lot of information. I still have a few more things to talk about in class, but hopefully you could answer the questions and you fill out your notes and come to class prepared with anything that you're still confused on. All right, I'll see you tomorrow. Bye.